background. So far in the class we have seen an important molecule called FOC1, and we saw how it was purified, and with the help of biochemical assays and crystal structures, its molecular working principles were understood. Now that we have seen that two monomers of FOC1 enzymes, for example, one monomer and second monomer, comes together to form homodimers, and this homodimers basically is formed at its cleavage domain. For example, if this is the cleavage domain, so homodimers are formed because there is an interface at around the cleavage domain site of it. And basically within the cleavage domain, if we talk about specifically, then you have aspartic acid 483, which forms a hydrogen bonds with your arginine 487. So these two are the hydrogen bond taking place from one monomer to another monomer. So these are the interaction surfaces. So that we have seen. And why this is important? Because in isolation, for example, the dimer of the cleavage domain can be utilized to link with any kind of DNA binding domain. That means you can link any of your DNA binding domain to target specifically within a gene and to link this cleavage domain at around that meaning slowly we are moving further towards the site specificity binding and its cleavage activity, because within the genome editing class, so what specifically we want from editing, or that we want to either insert, mutate, or you know change one amino acid or two amino acid, or within the base pair we want to actually mutate gene of our own desire at specific sites, whatever we want, only at that specific site that specifically change should happen, and nowhere else within the genome it should make any off target effects. So that we will see further in our classes. So one important topic of FOC1 we have covered, and we have seen the details that how dimers actually have one specific cleavage activity, and both dimers come together to cleave both strands of DNA. That means one cleavage domain cleaves one strand of the DNA, and other cleavage domain cleaves other strands of double stranded DNA. Now moving further, the second important topic is your zinc finger nucleases, right? So before seeing zinc finger nucleases, first we see that what are zinc finger proteins. So we will see in today's class what are zinc finger motifs, or what are zinc finger proteins, and how these zinc finger proteins were actually first identified, and from there onwards how it has been modified further for our own benefits in genome editing. So let's start today's story. So the zinc finger story starts with your Xenopus laevis, or any frog, Xenopus frog. So it was observed that this frog contains two types of 5S rRNA gene, one coming from somatic cells and one coming from egg cells, or oocyte type. So basically what you see here is 5S RNA gene. And now it was seen that this 5S RNA gene coming from somatic cells or coming from egg cells were differentially regulated throughout the development of your frog. So therefore, this system provides a good system to study regulation of gene expression and to study basic transcription mechanism. So this is how actually people actually first saw that these two genes are differentially regulated, and then they started working on this gene and found out several factors which were associated with this gene, and slowly they moved forward that in isolation of zinc finger. So we will go step by step, and in today's class basically we will see by what it was known by 1979 that the two types of genes, for example, oocyte type 5S RNA or somatic type 5S RNA, were both cloned and characterized. So what you see here are the figures where you have oocyte type RNA and somatic cells RNA. What you see here is the hybridization kind of assay where you first isolate total genomic DNA from Xenopus laevis, and then the genome was hybridized with 5S RNA transcript, and this 5S RNA transcript is labeled with H isotope. So that means you are doing a northern blotting kind of assays, and once the DNA was hybridized into nitrocellulose paper with your RNA, which is labeled 5S RNA labeled, you map where exactly your 5S RNA is coming into the nitrocellulose paper, and based on that you see a counting which is coming here. These are the counts per minute, and based on that you prepare a graph, and this is how you can see these are the somatic RNA and these are the oocyte RNA. So they were able to isolate this fraction and isolate this somatic cells, and they were able to clone and characterize these kind of gene. Now, another important advancement by 1979 was isolation of RNA polymerase 3. That was also purified and characterized properly. I am showing you just the picture where the different steps of purification was involved in your RNA polymerase 3, and what you get in the end is the specific activity up to very specific where it is utilized. So meaning that with these two system, one from the RNA characterization and second gene characterization, and second is the polymerase characterization. By mixing two, what you can do exactly in vitro experiments is something like in vitro transcription assays. So by 1979 or 1980, it was possible to perform in vitro transcription assays where you can have one gene. As you can see here, this is a plasmid. So this plasmid has your gene of interest. This is the gene of interest which is inserted here. And then together with that, you can mix your RNA polymerase 3. This is the purified RNA polymerase 3. And in the presence of appropriate buffer, as can be shown here in the this one. So when you mixed here, then at the end of mixing, you can see whether your gene of interest is giving rise to any RNA products or not. So this is called your in vitro transcription assays. So this kind of assays were performed, and it was observed that the purified RNA polymerase 3 failed to give any kind of selective accurate transcription when oocyte 5S RNA gene was incubated here. So there was transcription, but lot of transcription other unspecific products were also seen as I have described here. So these were unspecific. Whereas in the same kind of experiment, in contrast, when the RNA, same purified RNA polymerase 3, showed accurate transcription in the presence of immature or unfertilized egg. So this is the 5S RNA gene, same to earlier one. But when it was mixed together with unfertilized egg, you see a very specific kind of transcription going on. So this was the confusion that what a specific thing is going on. So with this kind of expression, they already see that there must be other factors apart from the purified RNA polymerase 3 that are necessary for specific transcription of your 5S RNA gene. So this was the whole experiment that we are going to see in today's class. So this is described very well in today's paper I have uploaded already. So this paper by David Engelke and Neg et al. In 1917 or 1980, what they showed exactly that they were able to purify a protein, namely A factor. So they have named this as A factor from oocytes. That means from cells, and these oocyte factors are important for specific transcription of 5S gene. What they did here, obviously I'm going to explain the purification step, various chromatography techniques, and then from there they went into complementation assays and how they were able to figure out this complete A factor, which was important for specific transcription of 5S gene. So let's see here. So what you have here, you can imagine this is ovarian tissue which was subjected to crushed, and then extract was prepared, and this extract was subjected to initial ammonium sulfate precipitation, and the fractions of ammonium precipitation were assayed for the ability of in vitro transcription assays. That means you have a gene which is cloned, 5S RNA gene cloned inside, then you incubate it with your RNA polymerase and together with your this ovarian extract. So that means also the ovarian extract will have some kind of endogenous RNA polymerase gene. So here is the first experiment when you mix this kind of assay. So this is the oocyte type 5S RNA gene mixed with your thing. So what you see here is specific transcription of 5S RNA gene. So this was good, but then when the crude extract was fractionated over DEAE cellulose column, and then you have the flow through. So from this flow through, you take out 24 microliter of this flow through, and then you add very small amount of zero point 0.1 microgram of your RNA polymerase. And when you add this two, then it was seen that there was no specific elicitation of any transcription. So that means the flow through, what is coming from this experiment, does not have any activation factor even in the presence of RNA polymerase. Therefore, it cannot elicit any transcription of 5S gene. Next, the eluate coming from the flow DEA cellulose was analyzed, and this, even in the absence of RNA polymerase 3, was able to give you a very nice 5S transcription. So as you can see here, this is with the LE. Although you have some background, as you can see here, these are some of the backgrounds coming from other reasons. So what they next did, they added within this eluate, they added next RNA polymerase 3. So this is the exogenous RNA polymerase 3 what they are adding. And after that, you see a complete black picture as you can see here. You can imagine there is a 5S RNA transcript here. But what is happening after the addition of exogenous RNA polymerase 3 is that basically it results in the addition of non-specific, I will say non-specific bands appearances. So that means lot of unspecific transcription actually starts by the addition of RNA polymerase 3. Now, when this whole cellulase eluate was passed on to this DAE Cefadex column, and this is done basically to absorb endogenous RNA polymerase 3. So you have endogenous RNA polymerase 3 bound here. And when you see this flow through, again you take out this flow through alone. Here it gives no transcription. Whereas when the same flow through was added with the exogenous RNA polymerase 3, what you see in both 6 and 7, you have very nice activity of 5S RNA transcription position. So you have some of the transcript made by this method. Now, when RNA polymerase 3 dependent Cefadex was again fractionated over different column, phosphocellulose column, then you have unbound flow through together with polymerase. It actually does not elicit any transcription, and this eluate, even in the absence or in the presence of RNA polymerase, does not perform any elicitation of 5S RNA gene. So this is no transcription is going on. So even in the absence of RNA polymerase 3 or in the presence of RNA polymerase 3, you do not see any transcription. But in the end, when they combine all three fractions together, that means you have flow through as can be seen here, then elevate and elevate in the presence of RNA polymerase 3. Then finally, you see some kind of transcription, but lot of unspecific unspecific transcripts are also present here. So basically from all these essays or for all these fractions, what they are going to conclude from this experiment. So basically they are saying that there is an existence of two or more components that should be required for 5S gene transcription, because in some of the components they give a proper 5S transcript, but in the absence of some of the components you do not see any transcription taking place. And at this point, I would like you to remember these two fractions, especially lane 5 and lane 9. These are coming from Cephadx flow through and one from the phosphocellulose elulate. And both of these do not have RNA polymerase, exogenous RNA polymerase. And independently, they are not able to elicit any 5S transcription in this case. So that means these two fractions are very good fractions for actually carrying out your complementations kind of essays. So now coming further, uh, they next studied the ability of unfertilized egg to accurately transcribe your purified gene. So this is your plasmid RNA, oocyte 5S RNA gene. And now they wanted to check whether this plasmid is able to give any specific transcription in the presence of unfertilized egg. Now, unfertilized egg, as we all know, is transcriptionally inactive. So they are not having any factors or all the factors required for any gene to be transcribed. But it was seen that although they are transcriptionally inactive, but their RNA polymerase is in equivalent quantity both in the unfertilized egg or in the oocyte mixture or the fertilized egg construct or ovarian construct. So what you have here, you have a prominent amount of RNA polymerase, but this is still transcriptionally inactive. So with this kind of system, that means individually this is also inactive. That means this extract can be used for complementation assays. So this is what they perform next. So they crushed this unfertilized egg, they made the egg extract out of it, and then they actually incubated the 5S type gene. So here the oocyte 5S RNA gene is coming from gene 3, that is why it is labeled as plasmid XBO X for Xenopus species B. Instead of Levis, it is coming from Borrelia and then gene 3. So this is the longer gene which is it is coming. So when they mixed these unfertilized egg with this gene, then they were able to see in the in vitro transcription that there was no activation. Obviously, because this is transcriptionally inactive, unfertilized egg, so you do not see any activation of transcription. There is no transcription of 5S gene in lane 1. But when the same extract was mixed with the lane 5, so that is this is the lane 5 I asked you to remember. So there is small fraction from the flow through, that is here, this was mixed together with your extract, then what you see, a nice activation in lane 2 from the unfertilized egg plus your complemented Cephadx fractions, that is your flow through. So when you mix these two, you see a nice activation of 5S transcript. Now, when you mix from lane 8, so this lane 8 was coming from the flow through plus exogenous RNA polymerase. When this was alone, it was not able to give 5S transcript. When this flow through together with exogenous RNA polymerase was mixed with unfertilized egg extract in the presence of gene 5S gene, what you see here, you do not see any specific activation of 5S gene. That means you do not have any fraction inside your phosphocellulose flow through, that means. And now coming to the interesting part, Then you have phosphocellulose eluate. So eluate alone is not able to give you the fraction, give you the 5S gene in the earlier picture, but when it is mixed with the egg extract, then you see a nice activation in lane 4 of 5S transcripts. So that means the eluate contains some of the fractions important for your specific transcription of 5S gene. So what you see here is a nice activation, and this is done with the help of complementation assay. And the outcome from this result, what we can predict from this, that the phosphocellulose eluate is missing in the unfertilized egg extract. So it has the RNA polymerase, but this does not have some of the transcription factor what is already present in the cellulase extract. So for example, in this eluate, something is there which is not present in the extract. Therefore, this either missing or it is inactive in the unfertilized egg extract. So the next job for them was to isolate this fraction. And before that, it is important that one kind of difference what you see in figure 1, if you say this is the figure 1 and this is your figure 2, the important difference between these two figures was the degree of selectivity of 5S RNA gene synthesis. For example, if this is the 5S RNA gene synthesis, it appears that the selective 5S RNA synthesis is much higher when unfertilized egg extract was complemented with your oocyte extract fractions earlier from the Cifadex or from phosphocellulose fractions, compared to the phosphocellulose fractions which was supplemented with RNA polymerase or exogenous RNA polymerase. So these fractions seems very unspecific. As you can see here, a lot of different backgrounds, but these are very clear picture and you see a only specific 5S RNA transcript going on. So therefore, the egg extract, they seem that the egg extract contain components of specific transcription of 5S gene from this whole experiments, right? So now they wanted to extract this specific factor, and for that what they did, they actually again fractionated the components of phosphocellulose eluate. So what you see here is the different fractions coming from for phosphocellulose eluate. And what they did here, this is the marker, as you can see here, MK. This is the input, so all the fractions what was there, and this is the flow through. So all the phosphocellulose fractions were eluated and precipitated with the help of ammonium sulfate. So then what you see here, the fractions from 1 to 16 were eluated in the presence of increasing amount of ammonium sulfate. So starting from 0.05 to until zero point 0.6 ammonium sulfate were used. And with this, what you see here is a precipitation of different kinds of proteins, and then you see different fractions. And what are the fractions? What you see? So mostly from fraction 7 to 16, what you see some kind of appearance of these proteins, and basically their putative transcription factor, they say, is inside this 37 kilo Dalton, and what they were unable to separate was it's a 34 kilo Dalton polypeptide. So this you see a very small amount of, even in the denaturing page, they were unable to separate these two separates. So now what they did, after taking out each fractions alone, first from lane 1, lane 2 to up to 16. So what they did, each fractions, from each fraction small amount like 3 to 4 microliters they took, and they incubated in the presence of 20 microliter of egg extract, that was unfertilized egg extract. So this kind of complementation assays they performed, and they are performing this to actually know that which of the fractions has the active component of transcription factor to activate 5S gene transcription. So here they are adding 5S gene, they are adding unfertilized egg extract. It will not transcribe only in the presence of the fraction which gives the transcription factor will activate 5S gene and it will give the 5S RNA. Right, so this kind of complementation assay they performed with all these fractions from starting from input to fractions to until 16 number of fractions. So what you see here is the result of complementation assay. For example, in the input lane, so what you have, the input is here. So when this 2 to 3 microliter was mixed here with the 20 microliter of unfertilized egg extract. So then you see a nice 5S RNA transcript. Whereas when flow through and until fraction number 6, when these were mixed in the presence of egg extract, you do not see any 5S transcription, no transcription. Whereas you see some kind of transcription appearance in or from 7 till 16 fractions. So this fraction you see a nice 5S RNA appearance. So with this kind of complementation assay, they were able to say that the transcription factor here, specifically the 37 kilodalton protein, is important for specific 5S transcription. That means this is the one transcription factor and they named it A factor. So that means transcription factor A is required, and they did another essays and our other publication support also. So they mean that this main important transcript is important for your transcription of 5S gene. So what is their next agenda? So for example, now they are able to see that okay, 5S RNA gene from oocyte is able to transcribe this 5S gene, and now they wanted to see whether this egg extract components are specifically important for 5S gene or it can also transcribe any other gene. So for that reason, they actually cloned, and this time they cloned the somatic cells 5S RNA gene, and then they prepared a plasmid harboring this 5S RNA from somatic cells, and then again they did an complementation assay with the egg extract, and as a control they also took tRNA. So here they took tRNA and then they performed the similar kind of assays. So what they see here, so when you mix somatic cells 5S RNA gene in the presence of unfertilized egg extract, then you see no transcription because obviously the egg extract is transcriptionally inactive. So you do not see any specific transcription at this position. Whereas when small amount of transcription factor was added, then you see a specific nice 5S RNA synthesis. So this is done in the presence of a small amount of transcription factor that they had purified from or that 37 kilodalton they had purified. Now the same experiment they did in the presence of tRNA that they had used as a control. So in this case, what they see that in the absence or in the presence of transcription factor, you still see tRNA synthesis. So that means this transcription factor A is not important for your tRNA synthesis. It is synthesized in unfertilized egg also. And even on top of that, even if you add transcription factor A, that does not hamper in any way in the synthesis of tRNA synthesis. That means tRNA or the transcription factor A isolated or purified is only important for 5S gene transcription. So this is what they did in this assay. And as a control, they took earlier ovarian cells, and from there they already had this control so that they can see whether their experiments are running properly or not. So this is the 5S transcription and these are the tRNA transcription here. So from this essay, they concluded that the purified transcription factor is essential for somatic and oocyte 5S gene transcription, and it is not essential for tRNA gene transcription. So this was their method. And finally, what they did next, that now they have isolated one transcription factor for 5S gene synthesis. Now they want to see where in the gene it is binding, because if it is a transcription factor, it will be binding DNA. So they, with the help of footprinting assays, they were able to identify the mode of interaction. And what they did here, they mixed the purified transcription factor with the labeled somatic 5S gene. And as you can see here, the control, they also incubated the purified proteins here. So transcription factor, purified transcription factor alone, or you have polymerase 3 alone, does not give any protection here in this lane 4, and polymerase 3 in the presence of unfertilized egg do not give any protection in lane 6. Whereas whenever you add transcription factor here, then in this lane 1 here, or in lane 3, or in lane 6, you see a specific binding or protection of the DNA starting from the region 47 till you have protection until 96 base pair. And these protection is inside your internal control region. So this was their outcome. And so basically starting from purification to complementation assay, in vitro transcription, then you have complementation assays, and finally with the help of footprinting assays, they were able to purify and see mode of action. That means mode of mechanism, how this transcription factor is working, and this is what they could figure out. So with this, what they conclude in this whole paper. So basically they are able to purify the transcription factor, and this transcription factor is specific only for 5S gene. That was demonstrated. The mechanism of action is very specific. That means it specifically interact with this kind of 5S gene to give the 5S RNA synthesis. The specific interaction is acting within this internal region of 5S DNA. So we will see here. So these are the internal regions of 5S DNA. And based on this, they actually propose a model that a factor provides the initial DNA recognition event, and its binding actually directs RNA polymerase 3 to subsequently interact with RNA and other transcription factors for forming transcription factor complexes, and it orients all together the whole complex to form a very specific initiation complex for transcription of 5S gene. So this was their observation in first paper. So with this, I conclude today's lecture, and this is now open for questions. So if you have any questions, then we will take these questions. And today we will again circulate the what you say forms, which will be open for from lecture number 5 till lecture number 8. So those questions, if you have, you can attend those questions. Yes, Kohilika.
Ma'am, can you please repeat the purification of A factor slide? I was confused in that.
Okay. Which part? So this one.
Yes, ma'am.
Okay, so what they did here, so basically from this experiment, we have seen that most of the components, for example, this component, right? So whatever is coming in the phosphocellulose eluate, right? So these are the component which is missing in unfertilized egg, because when you incubate eluate together with unfertilized egg, then you are able to see a very nice transcription product. That means something is there in the elevate of phosphocellulose, which is leading into the specific transcription of egg extract. This is clear? So now what they did, the elevate, all the fractions which are coming from the phosphocellulose, they actually fractionated from small amount from 1 to 16. For example, starting from input till flow through till up to 16 amount. They did that, they precipitated it with the ammonia, and then they actually saw which of these fractions are giving rise to a complementation assay. For example, if you fractionate this, then you get this kind of proteins. For example, here the proteins coming from 7 to 16 fractions, they were able to complement well for the 5S transcription. So that is how they were able to purify a specific transcription factor only causing for 5S gene transcription. Is that clear now?
Yes, ma'am. So we are comparing both the cases, right?
We are comparing both the cases. What do you mean by that?
That what's happening in case of the denaturing electrophoresis and complementation assay.
Yes, so denaturing they are doing because this is the cheapest way you can actually concentrate all the protein, and then you can fractionate in small amount and getting into, because those elevate might be coming in good amount. If you see the fractions, because they have not shown, if you see longer fractions, so there might be other proteins which might be coming on, but they might not be complementing the 5S gene transcription, right? So they are only
Yes.
showing the proteins which are complementing the 5S gene transcription. The protein which are responsible for 5S gene transcription.
Okay. Okay. Thank you, ma'am.
Yeah. Any other question? Aishwarya, please go ahead.
Uh, Ma'am, actually, I had this question that when doing the staining for the SDS paste gel, like, did they try with different methods like Kumasi blue or silver staining? Because silver staining tends to be more sensitive, so I had this doubt.
Okay, I have to see what is the timeline when silver staining was developed. I don't know exactly. So here in this paper, we are talking about 1980, and I don't know exactly. Maybe I can check and tell you. So I don't want to comment anything if I don't remember exactly. So yeah.
Okay, ma'am.
So what we have seen today, basically why we are doing this. So before there are two ways one can teach zinc finger domains. And if you see in YouTube's and lot of channels, there are a lot of content available basically on genome editing, starting from CRISPR, and there's lot of things on CRISPR, especially on zinc finger also there are a lot of things. But when you actually start understanding, there are not particular examples that how the whole field was developed, what was the reason, and why this protein is important. We know what this protein does, but why this is important and how actually it came into the field. So my idea of demonstrating this kind of example is basically two three kind of ideas I have. Because first, um, you will be familiar that how this field came into the picture. What was the reason that initially the zinc finger proteins were identified? And basically here in that respect, we are talking about transcription factor, right? So this is one way, like you know about transcription factor, you know about zinc finger. And the second important thing what I feel is, since you all are now PhD student and some MTech and prospective PhD students, right, in future. So basically when you enter into PhD, wherever in whichever labs you go, So your first idea would be to understand the topic. This everything starts with the literature survey. So if you are talking about one particular molecule, so A, for example, A. So you will have to read about molecule A, starting from what was it and how it came into the picture, then what is the present scenario about molecular A, and what are the open fields in molecular A, and how you will go further creating your own objectives out of molecular A. That means what are things not done on that protein. So this is why. So basically when you go about reading, then you will also come to know what are the methods by which people have proved. And sometimes I can also tell that sometimes what people prove, that via this method, one specific idea, that we can take an example of FOC1 enzyme, right? In FOC1 enzyme, based on the crystal structure, they obtained monomer crystal structure initially from Agarwal group, and based on that structure they actually deduced that there is only one catalytic site. But the question remained like how both stranded DNA are cleaved, right? But still they said, they proposed this mechanism that both strands are cleaved by a single catalytic site. But if you see today, from today's knowledge, that that proposition was totally wrong. And now we know that the dimer exists of FOC1, and that is how the both strands of DNA are cleaved. So similarly, in your case, if you go in researching your topic, then you can actually note down that what are the methods by which they have deduced this whole concept, and whether in today's time you have any advancement of these technique, like Aishwarya mentioned about silver staining, right? So same experiment could have been done either with silver staining or more sensitive assays, where one can see whether these kind of proteins are still holding true with those advanced techniques or not. So this is the idea, like if you can give increment knowledge, or maybe drastic total new idea to that specific subject. So that is why I'm taking individual paper. And also because you will be coming into the field, you will be taking individual papers, presenting that papers in your seminars or in your journal clubs, or you will be researching that paper. So you should have, should know the idea that how one paper should be selected and what are the different ways one can present that paper, right? So this is why I'm going through one and important. Okay, so today in zinc finger we have covered that how this protein was purified, and it was checked that 5s gene transcription is important, and this transcription A was actually studied. So in subsequent lecture, like tomorrow's lecture, we will see further like how this transcription factors were further went into detail, what are the scientists who involved, and how they deduce that this transcription factor is important, and why, what are the structures, and then why and how these are binding with the DNA. And why we are doing so, because specifically we want to create unique site within our gene of interest. For example, our human genome is carrying 3.2 to power 10 to power 9 billion cells, then sorry, genes. Then we need only one specific gene to be targeted. That means only one side, if we want to cleave, only one side should be cleaved, and there should not be any off targets. Off targets mean there should not be any cleavage to any other sides, so that if you also create any other site, that means mutations can happen to the other side. That means your whole idea of genome editing is reduced, or the efficiency of your editing will reduce over the period, over the chance. So our intention in genome editing is to find one natural substrate where we can modify those substrate of our own interest, create our own DNA binding protein, so that whatever site we want to actually mutate, we want to create new site, so those kind of experiments can be done. And this is done by slowly by zinc finger, and then talon, and then CRISPR. So slowly we will move further. So most important part within this whole subject was your FOC1 enzyme, because FOC1 enzyme cleaves non-specifically, and we have seen that. And in all these cases, FOC enzyme was actually linked with either zinc finger domains or with your talents or with even CRISPR. So these will be important, and we have covered in depth about FOC1. Actually, I have invested almost 7 lectures on FOC1, and subsequent lecture will be much faster because you already know what are the techniques we are talking about, footprinting and other old techniques, and specifically how the whole field developed. So we will talk about now transcription factor, and then further about talents. So with this is the idea of the flow through of the lecture, and I hope you are still pacing up with the lecture, like all the notes what I am giving. And your first quiz will be on month end, that is on 30th. So on 30th, what you will get in a quiz, you will get questions, for example, one word, you can expect one liner exam, and also concept based questions you will expect on that. And the topics what I will be covering in these quiz, first quiz will be your zinc finger proteins, that means the current topic, and second is your FOC1 enzyme. So based on these two, that means all 12 lectures, your quiz will come. Okay. So this is how you should go on and start preparing for the quiz and for other essays. Yes, Priya Raj, Anishi.
Ma'am, I
Priya
have two
Raj.
questions actually.
Yes,
So my first question is, we have carried out experiment with oocyte 5s RNA gene and somatic 5s RNA gene. But
yes.
in the case of somatic 5s RNA gene, in that experiment, we have used tRNA. Why is it so?
Okay, very good question. You tell me why it is so. Any
I
idea?
don't have an idea, and that's why I'm asking you. Like,
Okay.
what's the difference? Yeah.
Okay. So you know how many types of polymerases we have?
There are three types of polymerases.
There are more than three, but prominently there are three.
There are
Type 1,
three
type 2.
amino acids.
Yes. So what are they made for? Like polymerase 1 is for? mRNA is prepared by which method? Which polymerase? Sorry. Shawmik?
DNA Pol 2.
Yes. Similarly, polymerase 1 is made or is important for premature rRNA formation. Polymerase 2 is important for mRNA and other small RNAs preparation. And polymerase 3 is basically important for 5S RNA preparation and your tRNA preparation. So that is why you have as a control as tRNA. Like one of the transcripts for polymerase 3. So this is why they have taken tRNA.
Okay, ma'am. Ma'am, and my second question is like we have used different plasmids in both the experiment.
Yes.
Yes. So is that the reason like gene is same, but in the first case we took the oocyte gene, and second case we have taken somatic gene. That's why we have used a different plasmids. Is it so?
Yes. So we have used two different plasmids. I have not explained in detail, but you will see in the text. So these two plasmids are coming from two different sections. One from somatic cells. So you see one specific type of gene from somatic cells, and from oocyte you see three different variants of that gene, 1 and 3. So they have taken the gene 3, which is the longest one, and the orientation is probably different, as you can see with the star. So they have taken, that is why they have taken this prominent one. And if you read more about this 45S or 5S gene, you can see that there are lot of repetitions within these genes. For example, I can show you here. So this is one gene, and what you see here is internal control region. So this is just to show whether your transcription factor is binding to this region. And specifically the transcribed region has many more genes. So this is what your gene, then you have spacers, then your other gene, and then again 5s RNA gene. So basically this is repeated many times into the system. So that is how one gene is inserted. So you may so these are the part of actually probably earlier lectures. So that is why I'm not covering that in detail. But certainly, if you do not understand the basic thing here, you should go and maybe probably clear your concepts at that time, right? So this is why they have taken this gene 5s gene.
Okay, ma'am. Thank you, ma'am.
Yeah, yeah, welcome. Any other question? So by today, we will also open the forum for any questions pertaining to lecture 5 to lecture 8. So we will take all those questions. If everything is clear, maybe we can then close today's lecture. We have still 4-5 minutes to go, maybe 10 minutes actually, 5 minutes. We can conclude at 55. So in 5 minutes, if you have anything to ask, we can discuss about that.
Good afternoon, ma'am.
Good afternoon, Tirthankar.
Yes. So I had a question regarding the month end quiz and not the subject material. Can I
Okay.
ask that?
Yes, sure.
So the first quiz is on 30th. You mentioned that it will be single sentence or single word, basically MCQ types. So these papers,
Not necessary, but you might get the concept-based questions as well. Okay, not necessary that it will be only subjective or one word types. I will give some concept-based questions as well.
okay. So I was going through the papers and they convey a very long and complex story, and I get it at a conceptual level, but I'm facing a little difficulty understanding what I should actually prepare for active recall during an exam. So just could you guide me a
Okay.
bit with that?
Okay. So for example, I will not be asking anything like a mugging, but basically, um, for example, just take the topic of FOC 1, right? What is important in FOC 1? We have studied everything, right? So I presume what you said that it is difficult if you follow the lectures. I can assure you from my side that you will not find it very difficult. Although if you go through the research paper, you might feel little difficult, but if you go through the slides, you will still feel that things are clear. Okay, so especially in FOC 1, what you should remember, what are the catalytic domains, right? How it was found. Then what are the different types of experiments one can plan to, execute some kind of experiment. For example, how this dimers were found. Basically, they took some kind of recognition domain, they deleted the recognition domain, then they also mutated some cleavage domain. So these are the important experiments one can actually think of. And when you have any, some kind of experiments given, then you can also think like what kind of experiments to design for that particular notion. So these things will be important for FOC1 enzyme. Now coming to zinc finger, then of course not exactly the purification, maybe specific activity of pure proteins. Then white trypsin, where it is binding, what are the fragments coming. For example, in last paper, like FOC1 enzyme, there were 2-3 times similar questions were asked, that why after binding of DNA, two different kinds of trypsin fragments are generated. And this is a very valid questions. And this kind of question you can also expect in your examination, that if you are binding at a particular site, what happens to that particular site. And in my revision section, I have also mentioned that. that what exactly happens, obviously in very simplistic manner, that these are the prominent fragments which are coming and these are the prominent thing. So you can expect that out of all three or two topics, that what concepts you are gaining from those kind of experiment. For example, again, third example I can tell you, like footprinting essays, right? So sometimes you see in footprintings that complete sections are protected, and sometimes you see there is not complete protection of that site. So what is the reason for that? So those kind of question nobody asked till now. So I was expecting that question, but anyway, so those kind of things you can actually look for. Am I
So
a bit clear?
yes,
Or
ma'am. So basically, design of experiment-based questions and how they correlate to the story we have been going through in this coursework.
yes, a little bit of story and then design based question and analytical based questions as well. Like for example, if I give you the some gel picture, whether you are able to analyze that gel picture or not. So what kind, for example, if I in this complete, current slide, for example, I explained this figure, right? And then the same question was asked that why tRNA was used in this case, right? So the same kind of questions can be asked, like why certain kind of control was put in that particular experiment. So that will give you, for example, in DNA trypsin assays also some kind of control. So every experiment what I'm showing here has one lane which has a control. Even FOC1 trypsin digestion cleavage enzymes also that had one particular kind of control, and why that control was used. For example, in HINF1 digestion and FOC1 digestion, why HINF1 was used as a control. So those kind of things can be asked in the question quiz.
Okay, ma'am. Thank you.
Okay. So if there are no further question, I wish you all the best, and we will continue our zinc finger domains in next lecture. That is, we will meet on Tuesday then. Yeah. Okay. Thank you. Bye.
Thank you, ma'am.